All right, so people might trickle in, but I figured it's a good time to start the introduction. So yeah, so it's my great pleasure to introduce Luca Pagani. He's a colleague and friend, and I think I've known you maybe for 9 years. I think so when you were a grad student, sort of a
Yeah,
familiar face at the human genetics meetings. He's a very brash, and some of my labs described you as very Italian, which I think is
very
true.
Italian, yeah, definitely. No
Yeah,
secret
and
there.
so he obtained his undergrad degree in molecular biology from, I guess, the University of Pisa, and then afterwards went to Cambridge University, where his Ph.D. was in biological anthropology. Following Ph.D., he stayed in Cambridge for a while and worked at Wellcome Sanger Institute, University of Cambridge, and then went east a little bit to the University of Tartu, where he collaborated with Metz Palu and others, so last week's speaker. I'm not sure how much you're spending time, half and half, but at least when I last saw you in the flesh, you were sort of between Italy and Estonia.
Exactly. Yeah,
Um,
now it's like one week every month. So still, I mean, before the pandemic was 1 week
yeah,
every
yeah,
month. Now it's more of a, one off, like a
yeah,
month, a year, or something like that. But we'll see.
yeah.
Hopefully it's over soon.
And and since 2017, he's been at a tenure track position at the University of Padova in Italy. And I guess in March you received your promotion, so which is
Yeah.
fantastic. And one thing I think about is that in human population genetics, we've had this tradition of having um, important people named Luca. So you're sort of carrying the torch of Luca Cavalli Sforza. Big shoes to fill, but I think you're headed towards that. So it's great. When I think about the expertise that Luca Pagani has, I think of like being as one of the world's experts on the out of Africa migration, and you think about Northeast African genetics. And then finally, when I looked at his CV, I found that not only does Luca have expertise in population genetics, but he's also a little bit of a Renaissance man, and apparently you have a flute diploma. So musically
Yeah, yeah.
skilled as well. So with that, yeah, take it away, Luca.
Okay. Yep. Thanks again, Joe, for the invitation. It's a pleasure to be here. And before we were discussing also over email the target of this talk, and I don't know. I mean, in the end, I decided to keep it broad. So hopefully, it's a talk that people with or without a background in population genetics can appreciate. So let's see. Are you seeing my slide now?
Yep. And you might be
Good.
able to go full screen if you want, but we can
Yeah, yeah,
see
yeah,
you.
exactly. Yeah.
Yeah, it's good.
Okay. And so just to give you very shortly like an overview of the talk, I think the first half probably it's all about the rationale of the title. Digging ancient haplotypes out of modern human genomes. And because I think the main, the take-home message of the talk should be why, in my opinion at least, doing what is said in the title is the right thing to do. And then there are three case studies that shows, more or less successful examples of what can be done. Well, two of them are really pop gen and historical genetics in a way. The third one is more applied to the medical field. But we'll see. I mean, I'll go there little by little. The other important thing is that also Joe was saying I won't be able to see the chat, probably. But in any case, I'm sure and I hope at the end of the talk you will have questions and we'll have a discussion. But it's also very important that you are able to follow throughout. So if you have questions during the talk, both of out of interest or of understanding, if I wasn't so clear in some point and you want me to repeat stuff, so feel free to interrupt me. And maybe through the chat is not the most efficient way if I cannot see it. So either do it and hopefully Joe will flag it for me, or just open your mic and speak. I don't mind being interrupted throughout the talk. That said, I think we can start. So let's see. Okay, let's see if I can minimize this window. Okay. Cool. So as I say, let's begin from Adam and Eve. One important remark, I'm going to talk about Europe a lot during this talk, somewhat, and I'm going to refer to Europe as us, because typically I give talks in Europe. I realize this is one of the few times and I have a more international audience, so just forgive my Europe-centric view on some of the topics, but I'm sure you can understand. We're just talking about human populations across the globe. So let's begin from Adam and Eve. So just because, maybe you may not have, let's see why haplotypes are interesting, basically. Okay, so this is a simple representation of coalescent theory, and we just want to have this shared framework so we can understand what we are actually tackling. It goes without saying that my overall topic and interest is reconstructing the demographic history of human populations. And to do so, I typically use genomic data. Okay, so it is important, it's very easy to just these days to get some genome sequence, to get the data, throw them on a PCA, and try to wonder who's next to who and what that may mean. But it's also important to remember what is the the basic framework behind it, because that can help us making more and more powerful inferences. So just to cut the story short, here you get a set of individuals. This is the present generation, this is the past generation, and you can see that if you just postulate that the number of ancestors, if you just follow these females here, just female lineage, you can imagine a mitochondrial DNA. If you just follow the genealogical history of all the women present in the present generation here, right, you can postulate that the number of female ancestors of the previous generations was equal or smaller than the number of contemporary individuals. And that's very easy, as you can see it here. Hopefully you can see my mouse. It's just a way of defining the concept of sisters, right? This means that, of all the women alive in the current generation, some of them may be single child and some of them may be sisters, and hence they will share a mother. While some of the females that were alive in the previous generation simply will have no children, like this orange and this pink and so on. Okay, so that's quite easy. So if we all agree to that, then by definition, the number of ancestors will decrease or stay the same, or decrease as we go through the past until we reach the most recent common ancestor, represented here by this single female here. Depending on a lot of assumption and demographic events and so on, this event may take a few generations or very many, it depends, but eventually you will reach the most recent common ancestor. And so why is this important? And why did I put this Adam and Eve? Well, because if you know the story of Adam and Eve, and if you track mitochondria, you may get back to what is called the mitochondrial Eve, because eventually the first female who gave birth to all the maternal lineages around today, you can call it Eve if you like, but it's important to know that she was not alone. She was in a population that could have easily been the same size as of the one today. This scheme here doesn't need assumption of shrinking population to work. And if you do the same with chromosome Y, you can also reach Adam if you like, but there is no guarantee that Adam and Eve will have lived at the same generation, right? Because as we say, depending on the demography, the depth of this tree can change, can vary depending on many things. And so maybe Eve was alive 200,000 years ago and Adam was alive 300,000 years ago. But that's not the point. My point here is just that a specific part of the task I have in mind, that is reconstructing the demographic history of humans, is specifically comparing contemporary individuals or past individuals and trying to reconstruct what is the underlying phylogenetic tree, right? That is behind a specific locus, be it a mitochondrial DNA or be it a chunk of the genome, which I'm going to call haplotype from now on. So the easiest tree you can hope to reconstruct is something like, if I can go, okay, here. This is a quite famous mitochondrial DNA tree, or mitochondrial DNA is this DNA that we have in the mitochondria and that follow mostly maternal inheritance, and so it's a single locus. It does not recombine, and it's the exact perfect example of what can we do with simple tools to reconstruct a single phylogenetic tree. And that's why here genome, mitochondrial genome in this case, and haplotype is a synonymous thing, and you can just take the DNA as it is, it doesn't recombine, build the tree, and make some inference. Here, for your interest, it shows that our most recent common ancestor of maternal lineage was alive probably around 200,000 years ago, that the majority of the haplotypes the diversity is currently still living in Africa. These light blue and purple populations are all exclusively African individuals or individuals of African origin around the world, while everybody else, so all the other people who typically inhabit Eurasia, Oceania, and America, with particular reference to Native Americans, of course, keeping away the last 500 years of colonial history, they all are included within this tiny subset. But that's, this will be another talk called Reconstructing Out of Africa Migration. That's not the point, but still worth remembering that the vast majority of genetic diversity is still carried out by people who lives in Africa or who are of African descent, while everybody else in the world is just a tiny, tiny subset of the genetic diversity there. Okay, but again, the point here is that this is a very simple representation of human diversity because we're looking at a very simple genome, a single locus, mitochondrial DNA. Okay, but then what happens when you look at the whole genome? This is just a pedigree basically with a brother and a sister and all, father, mother. As you can see, if you just trace chromosome Y from the brother, then you're going to reach the father, paternal grandfather, and so on and so forth. And with the mitochondria, you can do the same and reach great-great-grandmother. But you see that already at the great-great-grandfather generation level, which is not so long ago, right? It's just 4 generations ago. It's 100 years ago, not so long ago. You already have 16 ancestors, of which you are tracing some information just for 2 of them. So you are deliberately neglecting 14 out of 16 of your ancestors. Grandparents. I even know the name of some of these grandparents, right? So it's not talking about Neanderthal. And what's important to realize instead when you use the genome, so nuclear genome, you get to see a contribution of most of these ancestors. Right? Not all the genealogical ancestors give you a genetic contribution for statistical reasons, but most of them do. And so it's much more powerful to look at genomes, and in fact, for the rest of the talk, we're going to explore genomes, nuclear genomes. But also it's much more problematic because, contrarily to mitochondria and Y chromosome, is not a single non-recombined locus, that actually the data you're going to get is a mashup of all these genealogical histories, right? So when you look at stuff, you want to be careful and you don't want to mix information which may derive from different phylogenetic trees. Okay, and in fact, the genomic counterpart of the simple phylogenetic tree I showed you before is something like this. It's basically a tree which contemplates admixture. I just chose these among many, admixture trees that are available in the publication just because this is nicely colored, and you have in blue contemporary populations, in red extinct populations, so either ancient humans or archaic humans, in this case Neanderthal and Denisova, and in green putative populations, okay? And the only thing I want to show is that in the previous tree you had your nice leaves that were connected by other leaves through simple, stems and nodes and so on and so forth. While here, some of the leaves, this leaf up here and so on, are not a simple connection to another node, but they are the result of an admixture. Okay? So, for example, here, the way to read this is that non-Africans are an admixture of 96% modern humans and 4% Neanderthals. I don't want to endorse this number particularly, but just to give you a sense. So things get more and more complicated when you look at genomes. You have to take into consideration admixture. Maybe it's an obvious statement, but it's important to remember that. Okay, so let's go to the main problem that we have here. If you just deal with modern genomes, and Joe and I were part of this publication in 2016 with a lot of modern genomes, each dot on this map of the old world, apologies for not having Americas, each black dot is a genome, and you can just throw a model there and say, okay, let's pretend that geographic distance between any 2 individuals, like this distance and so on, is proportional to the genetic distance. Or, well, the other way around, the genetic distance between any two individuals is proportional to their geographic distance. And the underlying assumption there is that people tend to marry more with people who live nearby. That's quite straightforward as an assumption. It may not be true at a small scale. It may make no sense when you compare people living 50 or 500 kilometers away from you, but surely when you start hitting 5,000, there is a higher chance of you mating with somebody who lives 50 kilometers away from you than 5,000. Okay? So it makes sense at the global scale. And so you say, this is my assumption, and so I calculate pairwise genetic distance, and this should pretty much correlate with geographic distance. And in fact, as you see here, when your colors are cold colors like blue or greenish, this assumption is met. When you see big red blobs, this assumption is not met. And as you can see, the assumption is not met just in some specific places, which are basically mountain ranges and a few other exceptions. So by and large, modern genomes can be described as, at least in Eurasia, as a huge uniform isolation by distance story, right? So the only reason why we are more or less similar in Eurasia is because of geographic distance. Okay, that's a very nice story. We know out of Africa, so you could hypothetically connect the dots and say, okay, so, there was the out of Africa. In the last 60,000 years, there was this equilibrium of, diffusion of genetic components, and that's all I need to know. In fact, if you inspect even a super small subsection of this plot here, I'm trying to center the square on Estonia. Maybe some of you may not even know where Estonia is. It's a very small country in Northeast Europe. This is Estonia, but to give you a sense, it's really, really minute. It's this peak over here in the Baltic Sea. So Estonia is a super small place with no geographic barriers. There are no mountains. There is no major river crossing it. It's paradise for let's just walk and go around and mate. And, even there, you can see some structure and some phenomena of isolation by distance. Each of these dots is an Estonian genome, and you can, as you can see, cluster them, with some effort, of course, but you can cluster them in orange, red, green, black areas, which more or less match geography, which again says that the only reason why there are southwest Estonians and northwest Estonian is because people tend to mate locally, and essentially it looks like it's a long-lasting history since the out of Africa, and people just kept mating and mixing their genes. However, we also know from ancient genomes that things were not exactly as simple as it may sound. This is a complicated PCA, and I'm not going to illustrate it in detail. If you're familiar with the ancient genome field, great. Otherwise, no problem. All I'm going to say is that the gray dots underneath this PCA are modern genomes. So if you just looked at the gray dots in detail, you may reconstruct the story I just mentioned. So long-lasting isolation by distance and people mixing with time and space. But actually, if you look at the colorful dots, which are ancient genomes, so the genome of individuals who died in West Eurasia, so these are just West Eurasian samples, and they died in the last 5,000 or 10,000 years, you realize that there are some dots like these yellow dots here and the red squares, more or less overlap contemporary individuals, and a straightforward conclusion is that maybe these yellow dot guys are simply the great-grandfathers of the gray dot guys. Okay, so that makes sense. Clearly, if you dig a hole, wherever you live, you find some bones, you find the DNA, there is a chance that these people, if they're not exactly your grandparents, they will be somebody related to the population who inhabit the land today. But if you see carefully, there are also other individuals, like these guys here, who do not overlap with anybody. And it's not an artifact, it's not a technical problem, is not an exception. This finding was replicated over and over in the last 5 years. And so apparently the evidence is that in Europe, there used to be individuals called the European hunter-gatherers who are not exactly ancestors of contemporary Europeans. To be more precise, this is also a very famous publication. The top part of the plot refers to contemporary population, the bottom part to ancient populations. We can just focus on the top part, it's not a problem. There are some familiar names here, French, Spanish, Greek, and whatever, Estonian, for example. Here, what I'm trying to say is that you could summarize the ancestry of contemporary Europeans into these three colors. Two colors, the orange and the green, would be immigrant, so population who arrived in Europe in the last 5,000 to 8,000 years. So in the grand scheme of things, Europe has been inhabited by humans for the last 40,000 years, right? So here we're talking about the last 10-20% of the time. While the other 80% of the time, Europe was mostly inhabited by these blue Western hunter-gatherer or other hunter-gatherers, but definitely not the orange and the green division. Okay, so what I'm showing you here is that in all the European population, you can see with some exception, you can see traces of these indigenous hunter-gatherers who were in Europe, but these traces are a minority. There are, maybe the maximum right in Estonian is maybe 35%, but it's definitely the minority. And so this means that when I was telling you this nice story of isolation by distance and geography, blah, blah, this story is correct, but probably doesn't really make us go back all the way to the out of Africa. Maybe, we need to dig further if we want to really know the specific contribution of the recent story, which is probably represented by the orange and green components here, and by the previous story, the hunter-gatherers in blue. And so the general topic here is that we know for sure that all populations, all contemporary populations result from the admixture of many other populations, right? Like this. So, you can see your favorite modern population as a mixture of population 1 and population 2, just generally. But like in the case of hunter-gatherers in Europe, when one of the two populations is missing because hunter-gatherers are not existent in the unadmixed form in Europe, and there is not even a population in Europe whose major component is hunter-gatherer, so you could use it as a proxy for hunter-gatherer. Before the advent of ancient DNA, we had no clue hunter-gatherer existed from a genetic point of view, and so we were only forced to see this, population 1 and the mixed population, and without a reference for the blue POP2, we were really lost, and we couldn't even tell that the mixed population was a mixture. That's the key point. And so ancient DNA can, save you and can come in the picture and say, now you have, I don't know, let's say a typical Middle Eastern population, which could be population 1. You have your European population, which you now know is the mixed population because you have an ancient population from Europe, which show you how mixed population is a mix between the Middle Eastern and the ancient DNA population, okay? Until here, I hope everything is clear. I cannot see the chat, but I hope you're still all awake and following. Okay, so key message now is if you have ancient DNA, you are king, you get the DNA, you reinterpret whatever we know of modern individuals in light of the ancient DNA, and we are sorted. End of the talk, right? Okay. However, ancient DNA is definitely a hype field and it's the way forward, as I just said, but it has a few problems. First problem is that, on the left side, you can see an Eppendorf full of modern DNA, and on the right side, you can see an Eppendorf full of ancient DNA. Striking differences are that the left Eppendorf, the left tube, has essentially DNA just from the target individual, the owner of the DNA, and the DNA is in very good condition. It's all long chunks. On the right-hand side, you have traces of the DNA of the dead individual in this case, here and there, and these traces are also smaller because the ancient DNA is fragmented. And to make things worse, you may have contaminant DNA, maybe the DNA of the operator in the lab, and a lot of environmental, mushroom, bacteria, DNA, and junk, right? Is that a big issue? Well, not if you are exceedingly rich, because this is the famous plot of how sequencing a genome was getting cheaper and cheaper until 2015, or even got a bit cheaper even more recently, but definitely the big drop was between 2008 and 2012. Okay, but if in your vial there is full and pure human DNA, then this is your reference cost. But if in your vial there is a 10% of the DNA you're interested in and 90% bacteria DNA or junk DNA, or 1%, so if your percentage of endogenous DNA is 10% or 1%, this is just this curve brought back 1 or 2 orders of magnitude, brought up. And so as you can see, your costs bring you back to 2009 or 2008. And so this means that to overcome this issue, you would have to pay 10 times or 100 times more for the same task, right? Which is, not a problem. Some labs can afford it. Then there are also some technical solutions like capture and so on. But, all in all, getting good quality ancient DNA is more and more problematic. And also, it's problematic, and it's more and more so if you go in regions like tropical area or Africa as a whole, where DNA preservation is poor, and so you may potentially not ever get ancient DNA from a specific part of the globe, right? So you don't have DNA, and your story is over. So the point, and this is almost the end of the introduction, but as I say, this is probably 50% of the talk, and this is why I'm making this story long, because this is the important message. One alternative or one complementary approach, again, I'm not advocating that you shouldn't get ancient DNA. That's the best way forward. If you can afford it, and if you can get DNA from the geographic point of the population you're interested in. But if you can't, either of the two things above, well, one could invert the perspective. So, we have a mixed population, which we know is mixed of population 1 and population 2, which you may not have available in the admixed form, but or you can hypothesize that the remnants of this population are present within your mixed, right? So you can see your mixed population here as a set of genomes. Each of these lines is a genome. And you can see this genome as a mixture of blue and black components, being black coming from population 1 and blue from population 2. And, getting the black components here would be boring and useless maybe because you already have your population 1 in an admixed form. But getting the blue one could be interesting because then, yes, with gaps, but you could get, unbiased bits in high quality grade, because this is no longer ancient DNA. This is the modern genome where you're looking specifically for chunks that you suspect may have belonged to population 2, to the blue population. And so, you can get this collection of ancient haplotypes from modern genomes. And so reconstruct what the ancient population looked like, or at least as best as you can using this trick. Okay, so this explains the title, Digging Ancient Haplotypes Out of Modern Genomes, because essentially is what we propose to do. It's not necessarily a new idea, and local ancestry has been used a lot, especially on many American populations, for example, African Americans or Mexicans even, where you have these very divergent European and African ancestries, or European, Native American, and African ancestries in the case of Mexicans, and then you can study the 3 components independently. However, let's say for African Americans, where you have European and West African typically components, both the European and the West African components are also available in the unadmixed form. So, of course, if you're interested in the specific history of African Americans and you want to ask questions like, where exactly from Africa or from Europe, can I trace these components to? Okay, that's a fair question, and that's the way forward. But if you want to ask questions like, how did West Africans look like, genetically speaking, or how did Europeans look like? Well, there is no point digging European chunks out of African Americans to do that. You can just go to Europe, sample a fair amount of individuals there, and then you get the answer, because Europeans in an admixed form are still available, right? Instead, if you want to ask questions of, how did European hunter-gatherers look like, or how did autochthonous South Indians, and we're going to see it now, Well, you don't have those people in an admixed form yet, so maybe this could be your only way forward. Okay, so I think that's the end of the introduction in quotes. I think we can have a look at some of the results. Okay, just one last thing. You could say, okay, sure, but then you get mutation. If an event took place 3,000 years ago, if 3,000 years ago the black population and the blue population mixed, and then now you want to convince me that you go into the mixed population today after 3,000 years, you dig out the blue chunks and you try to convince me that these blue chunks are witness of what the blue population looked like 3,000 years ago. There are mutations, there are things, and you're gonna be too different for your task to be successful. Yes and no, because here I'm just showing that after the admixture event, recombination takes place, and the recombination, and this we're talking about the red dots here, shortens your chunks more and more, right? So eventually you get a mosaic of the different ancestries, and mutation also takes place, right? But the trick here is that essentially, the more time passes, the more mutation accumulates, but the more your chunks shrinks, and so eventually you get, on average, just half or one mutation per ancestry chunk. So each of your chunks are going to look quite alike the chunks that were present in the mixing population, just because the mutation process is slow enough for you to have some good resolution 100, 200, or 300 generations after the admixture, I would say. But typically we work in a range between 100 and 200 generations, that would be safe. So ballpark 3,000, 6,000 years ago. Okay, so Bronze Age, which is not too bad. Okay, so one case, and it's quite schematic in this case. So here we applied this philosophy, this idea, to South Asians. And to the left, and this is a published paper, I'm going to cover these 3 case studies. They're all published, so if you also want to find more information, you can just look at the papers, or ask me, of course. To the left here, big gray panel, you get a PCA, and nothing new here. If you put East Asians, South Asians, and Europeans in a PCA, you get clearly East Asian on one side, up here, Europe on the other side, and in between, the famous Indian cline, which was first described by David Reich and colleagues in 2009, and essentially it means that you can see Indians, contemporary Indians, or South Asians in general, also Pakistani, and I think also Bangladesh is a good example. You can see them as a continuum of admixture between West Asians components, and East and South Asian components, okay? That's what it is. And so if you take, and the interesting thing is that in India, if you go in India and in Pakistan, you can find populations who are mostly West Asian, in Pakistan, for example. You will not find a population who are completely South Asians. So again, it's very easy to find the North component in an admixed form, so West Asia or North Indian component in an admixed form, but it's basically impossible to find the South component, so the typical South Asian component in an admixed form. There has been a couple of papers now on ancient DNA of South Asia, which was quite challenging to find ancient DNA there because of the environmental conditions. However, also in that case, there was no evidence, there was no genome available with just South Asian components. And so what we did, we took Pania, which is of all these many populations, the one showing the highest amount of autochthonous South Asian component, although it's admixed itself, this population. And we just extracted from all the Indians haplotypes, so chunks that looked like West Asian or North Indian, here in red, and haplotype that look like South Indians in blue. And as you can see, and so here these are just the insets. In colors, you can find the population, like Biror, Santal, Chamar, and so on, _N. This means Biror is an Indian population just looking at the north-like haplotype, and as you can see, predictably they fall somewhere else in the PCA, among other West Asian population. And again, this is the not interesting part, because if I was interested in West Asian haplotypes, I could just have sequenced people from West Asia. But the same population underscore S, meaning the South autochthonous version of this population, fall in an empty spot of the PCA, okay, as you can see here. And we did a couple of checks to show that this was not some artifacts or something. The interesting part is that probably they take the position which was originally occupied by this autochthonous South Asian before they were completely admixed to disappearance. So I think in this way we are showing for the first time what are the genomic features of these the people who inhabited South Asia before this admixture took place. This would be the complicated scenario showing all these many errors. This is from another paper, it's not from us, showing all these peopling of South Asia here and the formation of Indian cline. And we are showing that we are now probably being able to retrieve ancestral South Asian components, which you can put them in an admixture tree and show that this is the north component as a downstream component of West Asia, while the typical autochthonous south component is probably sister group of East Asia, or almost a trifurcation with Europe, East Asia, and South. And another thing you can ask is, okay, now I have these beautifully dissected genomes, and can I tell whether some particular SNPs are brought in the area, the alleles of some particular SNPs are brought in the area by the incoming population, or were there already, right? And here we are trying a simple way, just looking at frequency difference between the sources that we use for the ancestry deconvolution and the frequency difference between the ancestries that we managed to retrieve. And we see that there are some SNPs in this sector here, for example, SLC45A2, which are very differentiated between sources and very differentiated in the ancestries. And it looks like we can really pinpoint which ancestry is the one contributing most to this allele frequency in the area. But we actually have a more, in a way, sophisticated method, which is this one, which says, I can take all of my 25 populations, so maximum on the y-axis would be plus 25 and minimum would be minus 25, and ask, I know a specific population is, let's say, 50% North Indian, 50% South Indian, okay? And so I would expect, with some fluctuation, each locus of the genome to be more or less 50% North and 50% South. Do I find regions which are exceedingly North or exceedingly South? For example, 100% South or 100% North, or you can work out some standard deviation. Okay, so if so, I'm gonna annotate this region as a region exceedingly North, positive, or exceedingly South, negative. Okay. I do this exercise for all of my 25 population because, of course, I want this signal to be robust. And I annotate windows. For example, this window up here is saying that this window is exceedingly North in 23 out of my 25 populations. Okay. Another window like this one here, annotated with the gene SLC45A2, is exceedingly South in 23 out of 25 of my population. So while this fluctuation for a single population may be some statistical noise, when you see this signal to be consistent on all of your population, maybe there is something biological there, right? And we saw that indeed these two SNPs, which are responsible for hair and skin pigmentation, SLC45A2 and SLC24A5, are both quite popular if you are into skin pigmentation and hair pigmentation. While for 24A5, this pattern is not so clear, so these are all the populations we had available. And as you can see, so if a square is completely red, is exceedingly North for one component, or an exceedingly red is completely South for the component. And here, as you can see, the squares are not really decided in their trend, and some of them, like the blue ones here, are actually okay in terms of balance. But the SL345A2, the dot, is basically always completely red, meaning that it's consistently just South flavor, basically. And since it's this SL345A2 is involved in skin pigmentation and UV radiation resistance and so on, it makes sense that thousands and thousands of years of adaptation in South Asia could not be disrupted by the arrival of a not well-adapted haplotype from the north, right? So it makes sense. Okay, second case, I'll try to be quick, otherwise I'll run out of time. This is a simpler situation. I've studied Ethiopia for a long while, and we knew already that most Ethiopians, are the result of an admixture between, again, indigenous East African component and immigrant Middle Eastern component, okay, which arrived in the area around 3,000 years ago, following the so-called Semitic expansions. Okay, again, while in Ethiopia we have an admixed population, which can tell us a lot about the East African component that you can find there, we have no idea of who performed the admixture from the Middle East. And so again, you can apply the method and ask, how does the non-African component that you find within contemporary Ethiopian, how does it look like, and what can it tell us in terms of who performed these, migration and admixture in Ethiopia? So one way of doing that, and this is an outgroup F3, it simply essentially takes one outgroup, in this case Mbuti Pygmies, Central Africans, and then asks, if I take my reference population in Ethiopia, Amhara, okay, and a population X, which I'm going to list here, essentially, and I take my outgroup Mbuti, what is the genetic similarity between Amhara and X, okay, on the y-axis? And here it's saying, if you just look at this, it tells you that these populations here are the least similar to Amhara, and this population up here are the most similar to Amhara. But Amhara is an Ethiopian population with African and non-African components, okay? So that's my reference. Then I can take Amhara Naf, meaning Amhara just with the non-African haplotypes, so my extinct population that entered Ethiopia 3,000 years ago, and I can calculate the same statistic and see which are the population that most deviates from the the one-to-one axis. And so this point here, there were previous publications which were saying that essentially this particular population called Levant Neolithic was probably the most likely candidate for being the population who performed this admixture in Ethiopia. And so we pinpointed that, and these are the statistics for that population in particular, but we show that there are population, while indeed for non-admixed Amhara, this is one of the best population for these statistics, we show here that there is a huge amount of population who perform much better in being more similar to the non-African component of Amhara. In particular, most, if not all, of these purple dots, which are annotated as Anatolian. Now, the situation here gets more specific, but essentially, the question was, we know pretty much that the people who entered Ethiopia 3,000 years ago were belonging to either of these two groups, either Levantine Neolithic, so people from the Levant, meaning contemporary, Palestine, Israel, Syria, and so on, or from Anatolia, which is contemporary Turkey. Now, from an American point of view, this is probably, come on, it's the same backyard. True, but the people who were living there in that time were genetically quite distinct. So it's quite important to tell which of these two people performed the admixture. And performing this other F4 statistic, we can just show that while for full Ethiopians, Amhara in this case, Levant Neolithic and Anatolian Neolithic are equally good proxies for, who entered in the area 3,000 years ago. When you do this purification and you just look at the non-African component of Amhara for your statistic, actually for yellow and orange, these are some technical differences, but you can just look at the yellow. Actually, Anatolian Neolithic is a difference, makes a difference. And so you can actually challenge these two hypotheses and see that one of the two hypotheses is predominant, and this changed a bit our understanding of who were the historical population who might have performed admixture. And now we published this in last year, and apparently Sea people, if you're interested, might have played a role, or Philistines in a way. We can speak more about Ethiopia later if you want. Okay, so last example, and I hope I am in time. Yeah, no, I think 5-6 minutes I should be done. Okay, so I should say, so now we have seen two historical cases, South Asia, Ethiopia, and there we were interested in, we have admixed individuals of North and South Indian components, the first case, African and non-African in the second case. One of the two components is still available in unadmixed form, and I'm not interested because I could just study the unadmixed population. The other component is lost, so I'm interested in retrieving it, in digging it out of contemporary genome. Okay, those were two similar examples in a way. Now, what we're seeing here is a way of using the same tools, but doing something a little bit different. So this is the medical application. So now here we are interested in populations of mixed origin. And for that, the United States is great because there we see admixture of many continental level components, right? We get Native Americans and Europeans, Europeans and Africans, Africans and Native American with some European. I like these continental broad labels because broad populations are easy to tell apart, when it comes to ancestry convolution. I wouldn't be able to tell a Peruvian from a Bolivian Native American component or a French from a German European component, right? It's much easier to tell apart broad European from broad West African, East Asian, Native American, and so on. Okay, why all of this? Because we were already aware, and Kim et al., from your institute already showed that, and also other papers show that when it comes to predicting genetic risk scores for people, the broad population you belong to is very important. So, and there is a very specific reason for that. The huge bias in terms of which populations were used for discovery in the GWAS era. And clearly that for political and economical and unfair reasons is completely skewed, or it used to be, and now it's getting a bit better, but still there is a lot of work to do. The GWAS world is completely skewed toward individual of West European origin. This means that our understanding of genetic architecture, of disease and risk susceptibility is all or mostly biased towards Europeans. Okay, that said, right, the question is, we already know, and this was shown by Kim et al., and here I'm showing it again, that if you calculate polygenic scores, for example, for type 2 diabetes in this case, you can get fair, and you normalize for Europeans. These box plots here are normalized box plot of polygenic risk scores for CEU, which are Utah residents of European origin, Spanish, and Italian, Tuscans. Okay, and as you can see, you get some deviation, but more or less, as you expect, the overall population has a zero-centered distribution of polygenic score for diabetes. If you do the same exercise on individuals of Chinese or West African or East African origin, you get completely off, right? From this plot, you would conclude that all of your East Asian and all of your African samples are at high risk of diabetes. And I'm showing diabetes rather than height or BMI because this effect is much more clear in diabetes for some reason. And the interpretation here is that you can definitely not use betas or genetic effect size that were estimated on Europeans on other continental populations. Okay, that's known. And the only way forward to this is including more East Asian and more African and more Native American genomes in your GWAS cohorts. That's an ongoing procedure, and probably it will be achieved at some point. We all hope so, and that's the end of the story, okay? But the reason why we publish this is, okay, we know this big bias, and we know that if you are of African descent, there is no hope that we can use the European-derived betas to estimate your genetic effect size, genetic risk score. But what happens if you are 90% European, of European ancestry, and 10% African ancestry, right? What should we do? So, or is my performance going to vary according to your admixture proportions? But let's say if the majority, let's assume that the only beta we trust, and this is a bad oversimplification, are the European ones, just because the GWAS on Europeans are the majority. But also we have very good betas for East Asian. But let's just focus on European components for a second. And let's agree that I could do a nice job of predicting your genetic risk score based on your genome if you are of 100% European ancestry. Okay. If you are of 90% European ancestry, and 10% African, for example, or East Asian, what should I do? Should I just, remove you from, should I just prevent you from benefiting from this tool, or can I do something at least on that 90% of the genome, right? And so here we go again. What we did was, okay, I have my bunch of, so in this case, you have three cases. These were non-admixed populations, so 100% European, 100% East Asian, or 100% African. If I instead take, for example, Egyptians in green, Ethiopians, as we just saw, or African Americans, and if I calculate, just look at the green box plot, the polygenic score for these populations, as you can see, you get more and more off the more these populations have a non-European component. Right? Egyptians are, roughly speaking, 80% Europeans and 20% Sub-Saharan African components. Ethiopians are 50-50, and African Americans are like 80% Sub-Saharan African component and 20% European component. So you see, the more you get African in your genome, the worse I am at calculating your polygenic score for type 2 diabetes. But then we said, okay, but let's not throw this all away. Let's try and see what I can do if I calculate your polygenic score just for your European, in this case, component. And as you can see, for Egyptian and Ethiopians, you can retrieve a more or less zero-centered distribution of polygenic score, while for African Americans you can't, probably because the European genome that you have available is so little that essentially at some point noise gets beyond signal. And this is somewhat similar to what Joe did with ancient DNA, although this is just the first part. And then we say, this is just the distribution of polygenic score, but let's see what can we do if we actually, try and predict the R-square, so the association with phenotype. And as you can see, of course, we couldn't do much with European African admixtures because our understanding of African betas is still very poor. Things are advances quite swiftly, but back then in 2018-19, when we were doing the analysis, the betas for Africa were really not usable. But luckily, there was Biobank Japan, which was providing some nice betas for East Asian, well, for Japanese, but you can call them East Asian, broadly speaking. And so in UK Biobank, we had this bunch of European East Asian individuals, so individuals whose genome was partly European, partly East Asian, and we tried to predict for BMI, the polygenic score. We tried to predict the phenotype based on the polygenic score. And what I'm trying to show you here is that for European East Asian mixed individuals, if you try to predict their phenotype using East Asian dark green, that's your performance. You get an R-square of 0.02. If you pretend they're all East Asian, you get 0.02. If you pretend they're all European, you get 0.06, which is counterintuitive in a sense, but understandable because the size of UK Biobank is so much bigger than even if you get wrong betas, they are much more accurate than the right betas for a smaller sample size. But then, if you estimate partial polygenic score for calculated on the European chunk and on the East Asian chunk separately, and you combine them in this combined ancestor-specific polygenic score, you can get this purple distribution, which is, an improvement, a statistically significant improvement over just throwing at them UK Biobank betas, and it gets you much better than just using the naive East Asian betas. So just to summarize, and I'm done, the message here is that with local ancestry and applying ancestry-specific partial polygenic score to each of the ancestries, you can mix and match the different ancestries to create a combined ancestry-specific. And this has an advantage over just, you could say, okay, what's the problem? If I'm interested in African Americans, I'm going to recruit a few thousand African Americans, treat them as a population, and do a GWAS there. Nice. But African Americans, for historical reasons, are a pretty well-defined and abundant population. But these days, people can be an admixture of any 2 or 3 different ancestries, and you cannot hope to recruit a cohort for each specific I don't know, South Asian, European, South Asian, East Asian, African, whatever, Native American, East Asian admixtures, right? And you want to be more broad and maybe just learn on an admixed continental level population and then mix and match using this method. So with that, I'm going to shut up. And I'm sorry if I missed your question, but you didn't interrupt me, so I assumed they were not urgent. I'm gonna thank Padova and Tartu, for contributing to the research, and Mobilitas Plus, which is one of the funding agencies in Tartu, which nicely supports us, and a list of co-authors that you can see here. And with that, I think I'm done. Thank you.
Yeah, thanks a lot. It was a fantastic talk, really informative, and so I think people learned a lot of population genetics. And yeah, so we've got time for a couple questions. I'll just start with one and about the last thing you talked about. I mean, I thought it was really cool. I'm just wondering about when you're looking at sort of ancestry-specific polygenic risk scores, two related questions. One, how much are you trying to apply this to ancient European sort of sources in terms of that? Because you can imagine your mixtures of, steppe pastoralists and, hunter-gatherers and agriculturalists. And then also, does this method focus on like global genome-wide ancestry proportions, or does it use like chromosome painting approaches to say like, well, this part of your genome comes from this population as opposed to another one?
Yeah, so the second question is easiest, and it's local ancestry. So we actually say, you are European from position 1 to position 10,000, and the SNPs that are there, I'm going to use the betas for Europeans. Then the next chunk is African. I'm going to use African betas if only I had them, and so on and so forth. So yeah, it's more of a, stitch together different information and then combine them. But of course, so essentially, technically speaking, you first calculate the ancestry-specific European polygenic score. So you just look at the gaps, like you did for the ancient DNA, basically. Yeah. Right? And you normalize that, and then you do the same for the next ancestry, you normalize that, and then you combine the two partial polygenic scores. Second question for doing this exercise on ancient, if I understand correctly, if I have a European hunter-gatherer ancient genome, can I try and say what was the polygenic score for BMI of this guy, right?
Yeah.
I am not sure. I mean, it's doable, of course, but if you can see from this plot here, here to the left, okay, these are three modern European populations. So, they are more similar to each other than any of them to hunter-gatherer, and any of the UK Biobank cohort to the hunter-gatherer. And already here you can see that even though, so here we normalized on the full set of them. So these are 100 genomes each. We normalized on the set of 300 genomes. And already you can see that IBS, Spanish, are a good midpoint of Europe. CU tend to be protected for type 2 diabetes, and Tuscan seems to be at risk, a slight moderate risk for type 2 diabetes. And this is clearly not a real effect. It doesn't coincide with epidemiology. It's just the fact that what we observed at continental scale is also true at micro scale to some extent. Just to say that maybe hunter-gatherers would fall here, or super protected or super at risk, with potentially no power for us to comment on the actual usability of this, right? So my point is, yes, surely I can tell you the distribution of hunter-gatherers, but I'm not sure whether that would match what would be the R-square with the phenotypes.
Yeah.
And since, with the exception of height and maybe bone density, I would have no other phenotypes to use for hunter-gatherers. I don't know.
Yeah. That's cool. Yeah, so just wondering if there's any other questions. We have time for maybe one or two lingering ones as people head out for lunch.
Hey, hi. I have a question. Hey, hi. Fantastic talk. Very, very clear. Thank you. I have a question about the North Indian versus South Indian part of your talk. So there you talked about how you separate out the North Indian versus the South Indian component. And so how did you, so anything that was not West Asian, was that classified as South Asian, or did you use something like the Onge as a reference population or something?
Okay, so just to recap, Indians or South Asians today are a mixture of stuff that came from the north. It's confusing with the letters, but hence _N. And this stuff that came from the north is approximately West Asian today. okay, Iranian or something like that. And stuff that was already there, so real South Asian, it's difficult to say because South Asians are South Asians, and so we are talking about farther than South Asians, right? So called S. And for the stuff that came from the north, it was easy because we essentially just took Iranians. Actually, we took French, but also we did it also with Iranians, and it was the same. So imagine you have modern Indians, and you try to pull their genome to the French-Iranian cluster, and so if your haplotype is definitely coming from the north, it's going to be attracted there, and from the south, that's the question, right? And ideally, I would have loved to have pure autochthonous South Asian, which do not exist. We use Pania here, which are 80% autochthonous, right? And so that was the bet. We bet that even though Pania are not the right proxy population for South-South, when we are going to ask to an Indian, are you more like French or like more Pania? When the Indian is like the North, it's going to definitely have no doubt and go there. And otherwise, it's going to stick to Pania. And somehow, in some cases, it would be undecided, and in those cases, we just throw them away. But Pania, apparently 80% is enough for you to tell South from North. And the good thing is that we are not just biased, we are not just fishing Pania out of genomes, because as you can see, the South haplotype that we retrieved do not fall on Pania, but overshoot. And they actually go and occupy the right spot that you expected, where you expected them to find, which is somewhere intermediate between East Asians and Onge, Andamanese.
I see.
In fact,
Perfect.
apparently, which is from here, sorry, you can tell that the S component is essentially a trifurcation between S, Onge, and Han Chinese.
Interesting. Perfect.
There
Thank
is
you
just
so much.
2 units of drift, not making this a trifurcation, but essentially it's a trifurcation.
Very
So the
interesting.
autochthonous South Asian is not Onge, is a sister population of Onge.
Perfect. Thank you.
Okay, you're welcome.
Yeah. All right. Now, thanks again, Luca. And once again, if you have any other questions, feel free to email Luca, and we might schedule some smaller meetings a week from now. But once again, thanks again, and fantastic talk. Everybody have a great Thursday. Thank you.
You're welcome. Bye-bye. Talk to you soon.